All right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky, and this is Chen. This is part 2 of our ongoing discussion series on the topic of origins and ancestry. Now, part 1 was a discussion or debate, depending on how you want to look at it, between Donny and a tamed Chen. But this episode is Donny versus a fresh Chen, because you're rocking the fade. You are here
We're both
to
rocking the fade, Donny.
it's
Battle of
battle.
the fades.
It's battle of the fades in this episode, so I'm excited for it. And so, we're just going to get right into it because you know what, this is going to be an ongoing series, probably till the end of the world. Because my good man, we have a lot to discuss. And so, for those who watched part 1, we focus heavily on genetic similarities, chromosome fusions, and the chromosome 2 fusion in general. So we're just going to start where we left off, which was the chromosome 2 fusion. So let's allow you to bring up the first point.
Yeah, okay. Thanks for that, Donny. Thanks for inviting me again. Really looking forward to these conversations going forward. Very excited about it. And I learned a lot the first time I had a chat to you. So hoping that I can learn more and kind of really complete and fill out my knowledge going forward. Since the last week that we talked, Donny, what I thought I'd pull together was maybe more of a history on chromosome fusion too. I hope that, if your audience understands the history on where we got there, they might have a better appreciation as to why we evolutionists kind of say that it's a good evidence for common descent. Now, I'll state again for your audience.
We're entering history class now.
Yeah, history class, but it won't take long. It'll probably take 5 minutes all up, but I think it might be kind of good for you, for the audience, to kind of see the different years and a step it took to kind of build the picture that led to a conclusion of chromosome fusion too. So I thought that might be important. But I will kind of state again, all right, as we go through these multiple conversations, what I'm hoping to show your audience is that there are different parts and different facts, all right? When you take the whole facts into account, I believe, obviously Donny would disagree and possibly most of the audience, but I believe that all those facts, it's just easier and it's simpler that it points to common descent. But chromosome fusion 2 by itself doesn't say that common ancestry is more correct or creation is more correct. And I've said this in the first discussion we had as well. And Donny may or may not agree, but I can't use it to say yes, definitely creation is not true. What I'm hoping to show is that when we have, the second, third, fourth.
Are you basically saying it's a non-discriminatory or agnostic line of evidence?
As you say, it's agnostic. Now, I would think that it would be more likely. So then it becomes, you would probably say it's a worldview discussion or whatever, but I would say it's more likely that the evidence of chromosome fusion 2 points to common ancestry. But what I cannot say to your face, Donny, is that it would exclude creation, because as we talked about, there are scenarios that you can point to that I cannot dispute. You could say Adam had 48 and it can move to 46, whether it's the flood or whether it's pre-flood, as you said. I can't prove to you that did not happen. All right. So therefore it's agnostic. By itself, chromosome fusion 2 cannot say that your hypothesis is right or wrong. Okay. What I'm saying is that, and I'm basing it because I as we go on this conversation, we have parts 3, 4, 5, or whatever. To me, it builds a picture of common descent. So my head is already in that jigsaw puzzle where all the puzzle pieces are fitting together to common descent. Because the audience have probably only heard the first one, they may not come to that conclusion. I'm hoping as we kind of have these conversations, you'll eventually form that opinion. You may not. And this is why this conversation with Donny is going to be interesting, because it's going to be interesting as Donny gives me his views.
So in a way, it's a battle of the puzzle pieces. Do all those?
You said it was the battle of the fades.
Of the fades,
which is it, Donny? Pick a lane, mate. Pick a lane. Is it the fade or is it the puzzle piece?
You got some good barbers in Australia. I'm going to give your barbers some credit there.
Mate, when you've got a shaver, it's easy. You just go zoop, zoop, zoop. That's it, right? And then you just cut it straight, and that's all it is. Usually takes me 20 minutes to get a fade. That's the best thing about having a fade.
So with the puzzle pieces, those are basically your lines of evidence, your pieces of evidence. Yep. And are they fitting together to form a creation model or an evolution model? You would say the evolutionary model. I would say the creation model. And so that's largely what our discussion is about. This ongoing discussion is how do those pieces fit together?
And sneak peek, guys. And I'll be the first to admit it, and I'm sure most geneticists will admit it as well. As we kind of go through and talk about some of the other genetic elements, so I'm not sure we'll cover parts of it today or not, but I know we've got the expert in creationism view of ERVs right in front of me now, but we'll be discussing ERVs and the different other, even pseudogenes, etc.
I got to
All that.
send you a signed copy of my book on the topic.
Well,
Ian.
I've got your book, and
It's going to be worth
it's
money
not
one day.
signed. It's not signed, and I think it's the color version as well. All right, so is it the color version? I'll have to go check, but I remember
my autograph is very messy. It looks like a 5-year-old wrote my name, so it's probably a good thing you don't have an autograph copy because it would just ruin the book.
Mate. Ken Holman keeps on saying that a 5-year-old can understand creationism, so that's perfect then, right? So anyway, jokes aside, as I'm saying, and I'll be the first to say it, as we kind of go through chromosome fusion 2, and then we talk about ERVs, pseudogenes, or whatever other genetic elements, by itself, none of those things can point definitively to say creationism is dead. Because there's always some that you can come up with a scenario that could plausibly fit there. And so what I'm saying is, altogether, though, it's more likely. Okay, so altogether, it's more likely to point to common descent. And that's what I'm hoping to kind of show your audience as we kind of continue this.
And then, as you said earlier, obviously my position would be when it's all considered, then they would point to creation.
Wow. And this is where we will disagree, okay?
Right.
Because I'm saying it points to common descent, and you're saying it's but what I will concede is I can't definitively say that creation is wrong. But to me, so
now what are
in my mind,
your thoughts on this before you get into the chromosome 2 fusion? If you wanted to share slides or anything on that, Ian, feel free to get those up and get those ready. And so maybe as you're doing that, are you planning on sharing?
Yes, slides? I've got about 3 or 4 slides just on the timeline in history.
Okay, pull those up as I say this. And so the bigger picture is, and you kind of pointed to this, is a battle of worldviews, okay? And so I would argue that both worldviews or both positions, creation and evolution, we're going to be faced with pieces of evidence where we may have to derive what many would call a rescue device, right? But because we're coming at it from certain presuppositions and a worldview, then we're going to explain a lot of those pieces of evidence in light of that worldview, yours being evolution, yours being creation. Like we discussed largely in our first part, the Y chromosome dissimilarity, right? So on that specific piece of evidence for the creation worldview, it makes sense. Humans and chimps aren't related. Our Y chromosomes are only about 27% the same. For your worldview, it may appear contradictory, but then you're going to interpret it in light of your worldview and say, well, perhaps sperm competition, mating habits, faster rates of substitution, mutation rates in general could explain it. You get what I'm saying, Ian? Because you're coming at it from a different point of view.
And it was the first time that I heard, I guess, a scenario from yourself where you said that potentially the ape kind might have been chimpanzees and bonobos, and orangutans and gorillas would be, maybe a separate ape. I hadn't heard that before. And having that scenario does explain the data that we had pointed out around, why the Y chromosome wasn't considered between chimpanzees and bonobos, but I'm sorry, chimpanzees and orangutan, because they were quite more similar, but dissimilar, so to speak. So this is where I think as we go through the explanations, you will have a scenario that fits the data, and I'll admit that it fits the data, and I can't explain it. So yes, maybe you're right. Maybe the ape kind is 2 or 3 different ape kinds, and if so, that fits the Y chromosome data, better. All right. So I admit that. That's fine. So the only thing I'd probably say on the Y chromosome is, and you may call it a rescue device, but it's based on some evidence. So we know that Y chromosome, it has a lot of competition. So we would expect that there'd be high mutation on Y chromosome compared to the other chromosome, X or whatever, right? So we would expect that. The only what I then saw in the data, though, which still leads me to conclude common descent, was the differences between the different apes. Human, as obviously I consider an ape, so humans, chimpanzees, bonobos, and orangutans. The differences between them was still the same, whether it's X or Y. And what I mean by that is, when you just look at the X, the similarity between humans and chimpanzees was higher than it was between humans and bonobos, than it was between humans and orangutans, and humans and gorillas, and obviously the interrelations
What
between
are your
them.
thoughts on this then, Ian, in light
Yeah.
of those points? And I appreciate that.
Yeah.
If the Y chromosome, and it sounds like we agree, it is highly mutable. It changes more than your other chromosomes. It has a high mutation rate. Why then, in your opinion, in the evolutionary model, if we look at the Y chromosome in, let's say, just humans, we understand it mutates fast. We can debate how fast, somewhere between 1 and 3 per generation. Why are all Y chromosomes nearly identical? As in, there's incredibly low genetic variation in human Y chromosomes. But if the mutation rate is fast, and let's assume evolution for a second, so you go back to an out of Africa scenario somewhere between 100 and 200,000 years, people spread out in all parts of the globe, seize dominion over the planet, okay? So then you would have Y chromosomes worldwide, Chinese Y chromosomes, European Y chromosomes, African Y chromosomes. They'd be accumulating their own independent differences through mutation accumulation.
Yeah,
how come we don't find more diversity? Does that make sense, Ian? Does my question make sense?
It does, but that's if you only look at Homo sapiens by itself, right? So we know that if the Y chromosome is a essential part of reproduction, that anything that is not essential is likely to be the part that's being mutated and not fixed. All right. So I would hypothesize, and I say hypothesize because I'm trying to answer your question, Donny, in what I understand evolution to be, but I'm not a geneticist, so I don't know 100%. So I'll need to check this. But I would think that the reason why Homo sapiens and chimpanzees Y chromosomes are different is because they are both needed differently across the different species. So what's needed to keep a chimpanzee fit and healthy is going to be different from what's needed to keep the Homo sapiens fit and healthy. And whatever that is kept fit and healthy are the ones that gets fixed. Same thing with the chimpanzees. And they're just not the same because they're different species, different environment, and you would expect them.
But then what about just
humans? They would be the same.
Or you're saying humans, you would expect there to be low variation?
Because it would be, so I mean, I'm not sure how much of a Y chromosome affects things like ability for cold weather, but I would think it'd be something around maybe intelligence, around, things that we know that all humans share. That chimpanzees.
So with those mutations then in the Y chromosome within the 100 to 200,000 years since the out of Africa scenario, are you saying a lot of those mutations are weeded out through selection? Because if they're accumulating,
well, they're not fixed. All right. So it would either occur in the places that is not essential for Homo sapiens, like what it means to be human.
Okay, let's say they're not fixed then.
Yeah.
What about these subgroups? So you can still have unfixed differences. So let's say you compare different people groups around the globe. Let's say African people groups, Chinese people groups in the Y chromosome.
Yeah.
How come they're not more divergent? Why isn't there more how come there's so much uniformity in the Y chromosomes if they're so rapidly changing? If evolution was true, I'd expect more divergence in Y chromosomes globally.
But that's what I'm saying. I'm saying if you are just looking at it in terms of humans itself, that they probably are, compared to an X chromosome, relatively more divergent, right? But if you're comparing that between humans and chimpanzees, then I would think that the divergence between those two would be even bigger than it is to be within humans itself. Does that make sense?
Well, the X chromosomes
you're making it sound as if an Asian person and an African person would have this huge differences in Y chromosome, where I'm saying, no, a chimpanzee and a human, you would expect to have a very big difference. Now, it's surprising that it was 27% similarity. I don't think people expected that much difference, but you'd expect to have a big difference. However, you wouldn't expect that big difference between, let's say, Asian and African. But what I'm saying is that I'm sure if you look at
Asian, African, so take any two Y chromosomes on the globe, compare them. They're nearly identical. You can actually take human Y chromosomes, map them out on a family tree, and they go back.
Are they as identical as, let's say, the same thing on the mitochondria chromosome, the X chromosome? Are they? All I'm saying is there's the relativeness is what I'm comparing. So
your mitochondrial DNA. It's a good question. Same thing. Very low genetic variation. Nearly, I think in the mitochondria. So the mitochondrial DNA is smaller, right? The compartment's about 16,500 letters.
Yeah.
And on average, any two people in the mitochondrial DNA differ by about somewhere between 25 and 30 differences. Your
Yeah.
max is about 100, 120. Your Y chromosome is a much bigger DNA compartment. I think it's about 65 million DNA letters,
Yeah.
but the average is still very small. It's probably a
Yeah.
few hundred. So
Yeah.
overall, you have low variation in mitochondrial DNA, low variation in Y chromosome DNA, which fits an origin of the mitochondrial DNA and Y chromosome of, let's say, 4,500 years. But if they do go back to an out of Africa scenario, roughly 100 to 200,000 years, and the mutation rates are high in both those DNA compartments, my point is, Ian, I would expect more DNA differences. I would expect more variation over that extended period of time. Does that make sense? And what are your thoughts?
It does, but I guess because I'm trying to look at it in terms of all ape species, because remembering I come from a position where
Where
humans
they're all related.
one, yeah. So my question then would be, are we seeing the same level of similarity within a chimpanzee Y chromosome? So you've got, I don't know, you've got, what is it, 2 or 3 different species of chimpanzees, right? You've got at least 2
More
species of
diversity, I think.
yeah, like I would think that within chimpanzees, there is less diversity than it is between the chimpanzees and the gorilla. Same thing that we see with humans. I think there's 2 species of, or there's a there's the yellowback gorilla or the high plain or whatever they call it. There's at least 2 or 3 species of gorillas as well. Same thing. I would have thought the Y chromosome within those species would be more similar than it is between that and an orangutan. Do you see what I mean? So if that logic still holds within each of the different species, Homo sapiens, the different species within orangutans and gorillas and that, then it makes sense that when you're going back millions of years, because that's when they diverge, you're going to see the differences between an orangutan and a gorilla and a human being much higher than within the different species itself. So to me, it still fits the evolution worldview in that within the species, maybe it's not enough time. Maybe we need millions of years. I don't know. They say modern humans is only 200,000 years old, so it's far shorter than a 6 million split that I think they're saying is the common ancestors between the chimpanzee
And so and I appreciate what you're saying, and so you make some fair points, but right there is the issue that I have.
Yeah.
Is modern humans, according to your model, are somewhere between 100 and 200,000 years. Okay, because you would put Y chromosomal Adam somewhere in Africa. Okay, and he would have belonged to a population of men, according to your model. For me, it's Y chromosomal Noah going back 4,500 years. Okay. So if you're accumulating 1 to 3 mutations per generation in your uniparentally inherited DNA compartment, which is autosomal it's nuclear DNA, but not autosomal DNA, the Y chromosome. And it's a smaller DNA compartment, right? 65 million DNA letters. Then those mutations over that amount of time should result in more DNA differences or DNA diversity in just human Y chromosomes. Then I understand you bring in the chimpanzees, there's even more differences. Actually, within chimpanzees, there's quite a bit of differences, just the way they behave.
How many genes are in the Y chromosome? Do you know?
Genes, I'm not sure. I know the amount of DNA letters is about 60 to 65 million.
All right.
Compared to
So you have a
autosomal
mutation
DNA, you're
in one
looking at
letter. So when you say there's one mutation, is that one letter that's changed?
Yeah, well, you could have deletions. You could have
So
there's different mutations, right? But the thing is, it's also, we talked about this last time. Your Y chromosome is non-recombining because it's not autosomal DNA, right? There's no chromosomal counterpart to recombine with every generation. So it would be more stable in that sense, but you could say it's less stable in the sense that it's accumulating more mutations without recombination, or maybe a little recombination or gene conversion at the tips, they say. But what we could do is park that just so you can get to your chromosome fusion.
Yeah, let's park it. Sure.
Okay, let's do that.
Okay. Yeah,
because I do see your
like
slide.
I learn stuff when I chat to you, and I'll have to go and have a kind of, I'm not an expert, like I said, on.
No, this is great.
But it's interesting.
I like these because we get to dig deep.
Yeah.
And before we do each next episode, we get to go back and study these things even more.
Yeah. And I learn and you learn, and it's wonderful. All right. So all right. So I just want to give the audience a bit of a history and hopefully an appreciation as to why I think chromosome fusion 2 points to common ancestry. And I think a part of it's because of how we discovered it, right? So I'm sure Donny will then go into some of the genetic elements of it. So this is really a more of a focus on the historical part of it. Okay, so a bit of history. Is that all right? Okay, so just a bit of history lesson, guys. So Tijo and Levin in 1955 discovered that humans had 46 chromosomes. Now, previously, it was assumed that it was 48, and that's because the year prior, I think 2 years prior, they had found that chimpanzees and gorillas both had 48 chromosomes. And I know a lot of creationists here would like to argue differently, but the discussion around common ancestry was settled, a long time ago. It's nothing that scientists kind of debate today. So it was automatically assumed, well, because chimpanzees and gorillas had 48, that humans would have 48 as well. And surprisingly, in 1955, when they did kind of look for chromosome counts in humans, they found 46. So this was a problem. So this was in 1955, and it became a kind of problem at the time to try and explain why is this different. So a number of papers have come out since then. And keep in mind, the technology to compare chromosomes is not as high resolution as we have it today. Now we can actually measure the different amino acids that are there and count them and kind of compare them. Back then, we didn't have the technology to do that. So here are 3 different papers across 1962, 1963, and 1969, where they used different methodologies to kind of compare the chromosomes between humans and that between chimpanzees. So the one by Chiarelli in 1962 used the way of kind of measuring similarities in length and different mathematical criteria to compare how similar they were. And they concluded that, well, it looks like some sort of fusion happened. Now, keep in mind, it's not high resolution data, but they were looking at lengths of chromosomes, and they said, well, this is roughly the same. So we think that these 2 chromosomes might have been put together, and that's why 48 became 46. And they were basing it on kind of length of the chromosomes that they could see, and a different kind of indices, different kind of potential chemical signatures that they can kind of match. So that was in 1962. In 1963, a further study was done with, back then, a bit of a newer technology where they could measure some of these indices, these chemical signatures, and kind of match them better. And they also concluded that if you match these chemical signatures across the chromosomes, it looks like there was a chromosome fusion that occurred, and that's why 48 became 46 in humans. And then in 1969, they did another methodology where they actually use a chemical to kind of stain the different chromosomes, and they compared the different staining of chimpanzees and gorilla chromosomes with humans, and they also concluded that if you compare the staining patterns, it looks like there was a fusion. So it was a surprise in 1955 that humans have 46. There were 3 different methodologies, like I said, not high resolution methodologies, but even the chemical staining methodologies and the kind of comparison on the different length of chromosomes seemed to point to a fusion. But the big study that was done was in 1972. Chiarelli, who did the 1962 study, and they basically did a comparison of chromosomes. And this was the first time where you were able to kind of stain, or what they call band. You guys, I have a photo of it. Actually, I might have a photo here. No, I have a photo of it on the next slide. But if you guys can kind of cast your mind back to maybe biology in high school, and you could see the chromosome and the different staining of the banding of the chromosomes. This is the first use of the technology to get a low resolution view of that. And when they kind of did the banding staining of each of those chromosomes, and they were able to compare the different bands, they pretty much could see a very, very similarity between 2 chimpanzees and gorilla chromosomes matching to a longer human chromosome. And they kind of concluded that a fusion is most likely to have occurred. And they hypothesized then that
Ian, if I could, that staining.
Yep.
And I appreciate this history here. It's good information. The staining is karyotyping.
Yes, correct.
Yeah.
Yes. And keep in mind that the papers that I'm showing as we go over the years, that staining becomes higher and higher resolution. So this is the first time they did staining. They call it banding, and I think they use some like there are different types of banding. I can't recall what this one. I think this one was, and I've got the papers there for everyone if they want to search for it. Most of them are open access, especially because they're quite old. So you can read them.
Pretty cool because they can actually isolate cells, Ian, as they're dividing during mitosis.
Yeah.
And so the staining that takes place gives you a visual picture of the
Yep,
chromosome. Yeah.
Yes, correct. And you can see I've talked about the karyotypes here as you pointed out. So you understand this, right? So a prediction was made basically. So they could see the staining of 2 chromosomes, and when you match those, you put them together and you match it, it matched chromosome 2 in humans using the banding. Now, I will state again, this is the first time that they've done this, what they call banding, the staining, they call it banding. It's the first time that they've used banding, and it's not as high resolution as we have today. Today, it's so high the resolution that we actually measured it 4 different amino acids. We don't even stain it. We know what those acids are. CATG, we know what they are, and that's what we compare. But back in 1972, we didn't have that technology, so it was just a visual comparison. And this is the first time where...
Yeah, can I ask you one question based on this?
Yeah.
So from 1955, they discovered humans had 46 chromosomes. So previously they assumed there was 48.
Yep.
How long after that, and I'm looking at your other sources,
7 years
is that when they made their prediction that okay, there must have been a fusion to account for the
specific prediction was in 72, but about the data that came, so the question was, oh, that's strange, why does humans have 46? This is in 1955. We've done it for chimpanzees and gorillas, and it was 48 last year, so we just assumed it was 48, and now it's 46. There's something funny going on. So they did it. So there's 3 key papers that came out listed here. Excuse me, guys. Just let me get some water. So
because of your fresh fade, we forgive you.
Yeah, it wasn't so much a prediction. It was assumed, all right, that there had to be some sort of fusion event,
right?
Because like I was saying when you got dropped out a bit, was common ancestry was something that's been around for over 100 years. It's not something that was debating back then. I know we're debating it now, but it was accepted even back in the 60s. So then the question was, why is chromosome different? They did a bunch of studies. They used chemical analysis. I'm more like measuring the different lengths of it wasn't exact, but that's what they did. The 3 different studies in the 60s show that if you use those methodologies, it looks like there was a fusion. It was in 1972 where with the banding, that was at that time the best and most accurate way to show visually that there was a fusion that occurred. And a prediction occurred. This is the first time the prediction came up because they didn't really know about what made up a centromere or telomere. They didn't really know. They knew kind of that there was these end caps to a chromosome. Didn't quite know what it was made from. So there was a prediction that if there was a fusion, those 2 end caps would have had to connect, and so there would have to be a telomere fusion site. Okay, so that was one of the predictions that came out in 1972, that if this is true, we should be able to see it. They didn't have the resolution to see it at the time, so it was a prediction that said we should see a telomere and telomere fusion site, but we should also see 2 centromeres, because each X chromosome had its own. If you fuse it, and you'll see the picture here, you'll see a centromere on each one here. If these 2 are to fuse together, they would have, chromosome fusion 2 would have 2 centromeres because it's a fusion of 2, and it would have a telomere fusion site because that's the telomeres are the end caps that kind of join. So those are the specific fusion. They could not see it at that time with the banding technology, but that was the first time it was predicted that would be what we should be seeing. Does that make sense, Donny?
Yeah. So basically, you'd expect if your chromosomes were to come together, fuse together with your end caps being telomeres, the telomeric repeats, you'd expect to find in the center of that chromosome a good amount of those telomeric repeats, and as you mentioned, an actual centromere and then a silenced or cryptic centromere. Basically,
they didn't know about the so back then they didn't understand what is it a centromere did. Now we know that a lot of it's around the stability of the chromosome. So they didn't predict one of them would be silent at that time. They just said we should see 2. Okay, So they didn't realize that if you actually had two that and both of them were working, it would actually be a big detriment to the chromosome. So obviously, going forward, you would have to have one of them being silent. But at that time, they didn't know about what a centromere and what its big impact is to a chromosome, but they predicted that there would be two. Okay, so yes, to your point, you're right. One of them would have to be inactivated, but they didn't know the importance of that until later on, if that makes sense. All right. Okay, so that's prediction one. So we go to the next one. So 1973, Turlio and Grouchy, they did probably there was a much newer technology around banding, and this was the first fairly high resolution banding. And these are screenshots from the actual paper that they kind of published, so you can see what when Donny and I talk about banding, it's a different shades here that they kind of see and they kind of match. And you can see that they compared it between chimpanzees and humans, so Homo and Pan on the one on the left here, and also on the one on the right, specifically, what the chromosomes kind of look like. So you've got the human in the center, so that's a human chromosome 2, and you got the two different chromosomes for chimpanzees on flanking either side. So when we say we compare the different bandings, it's basically looking at the different bandings on the long chromosome and seeing that they match very similarly to the two different chromosomes on the chimpanzee side. So this was the big paper in 1973 that basically went, oh, wait a second. This 1972 paper said that this is what would happen, very low resolution. 1973 published a more detailed, high resolution of this, where people could look at it and kind of go, that's really strange. It looks like there's a fusion based on the banding technology, and they call it R banding here, the R banding technique. I think the previous one was called the B banding, so it wasn't as good. But this R banding technique that's used in this paper, people could actually see now for themselves. It's pretty similar. So, and this will be the story as we kind of go on. Each paper that gets published,
with that specific point, just to clarify, that specific point is because of the similarities in the banding patterns, the homology or the synteny?
Yeah, so that they looked at. So remembering, we did not have the technology to figure out the CATGs that sat in the chromosomes yet. So it was all visual. This is 1973. It's good, 50 years ago still, right? The technology. But I'm trying to build a picture on, as we get more papers with better technology, all those papers confirm what was the conclusion previously, right? So like I'm saying, all I'm showing you is evidence that chromosome fusion occurred. What I'm not showing you is evidence that therefore creation is wrong. Okay, I'm just saying that for people that deny that chromosome fusion, like Ken, I think his rationale is, I don't know that a fusion occurred. I mean, okay, whatever. I'm saying you just cannot deny that a fusion occurred.
What's the biggest point that you would say is conclusive data that a fusion occurred? Because we do have the banding patterns, the homology, we have the synteny, but all of those are largely agnostic. What would be the conclusive point or two that says, okay, this was definitely a few? Because as you said, the two big things we'd expect that would point more towards a fusion happening would be your telomeric repeats and then your cryptic centromere. I would say the homology and synteny can go either way, common design or common ancestry.
And at the very beginning, even in part one of this discussion, I said to you, chromosome fusion 2 does not point one way or the other. Okay, it's a whole bunch of data we'll go through as we go through this series that I believe will point to common ancestry much better, right? All I'm trying to address here is some people in the live chat comments that I kind of read through still dispute that fusion occurred. What I'm saying is you can't, unless you are stupid, and I'm not saying your audience is stupid, but maybe, unless you were really dumb, you can't deny that a fusion occurred, right? And by the way, this is the same way we know a fusion occurred in deer species, in muntjacs, in any other species, like even dogs, wolves, and foxes. They have different chromosomes, right? This is also how we know a fusion occurred. This is the same methodology, right?
Right, so but I'll say this, Ian. See, so if you would look at my screen briefly, okay, and I appreciate that history. So here's my issue, okay? I agree chromosome fusions happen. We have examples of them happening in nature, in the wild, okay? We have examples in pigs, equids, deer, so on and so forth. When we look to the purported fusion site in humans, in human chromosome 2, okay? And this is largely what you went over, so I'm not going to belabor the point. but as you can see here, I do like this visual because it helps us to understand what we would be looking for. Okay. And so as you put it, you'd have your ancestral chromosomes, right? They called them 2A, 2B in an ape-like ancestor. So here's your centromere at the center of the chromosome. And then you've got your end caps, which are your telomeres.
Yep.
Those comprise DNA letters, TTA, GGG, CCC, TAA. Okay.
Yep.
And so these come together. Correct me if I'm wrong. So these come together.
Yep.
And so now you'd find an abundance of telomeric repeats here. Obviously, you'd still have your telomeres at the end, and then 2 centromeres. Okay, so here's the prediction. So everything you put, and here's the staining that you went over as well, the karyotyping, which is very good. So here's my issue. So here's what we would expect to find. So here's the chromosome 2. So we should find the telomeric DNA sequences here in the middle. You should find 1 active centromere and then 1 inactive or silent centromere with your actual telomeres. Okay, but so here's where my issue is. So in your examples of fusions, so we have an example in a certain pig species. The signature is huge. It's thousands and thousands of DNA letters. I think in the pig fusion, it's 50 to 60,000 DNA letters long. You have many tandem repeats of TTA, GGG, TTA, GGG. Okay. But what we find, so that is indisputable. I agree. Those fusions in various equid species, a pig species, they're indisputable. The signatures are large. They're there. They can be seen. Okay. But here's the purported fusion site in human chromosome 2. Rather than it being what we see in these various animal species, Ian, it's very small. It's less than 800 DNA letters large. Okay.
Close, 798. That's your memory's quite good, Donny.
A little bit less.
Great memory. It's exactly 798. So you've got it spot on. Yep.
So 798. There's really only a few. So here's your TTA, GGG, and it's very degenerate. So what that means is, notice this, TTT, GG. So you don't get another tandem repeat until many DNA letters.
So I've got a view on that one. If you, I think maybe on the next slide, if you can put up my slide again.
Yeah, so let me do that, but I do want to, okay, so I want to point this out, and then I'm going to put your slide back up.
Yep, of course.
This is from a prominent paper on this issue, 2002, fan-wide, genomic structure and evolution of the ancestral chromosome fusion site. They said this, because you make a good, see, the gold standard of science is making testable and falsifiable predictions, and then testing those predictions and seeing if they come true or not. Hopefully they come true, right? If not, go back to the drawing board. So they said this: if the fusion occurred within telomeric repeat arrays less than 6 million years ago, this is not a creationist asking this question.
Yep.
Why are the arrays at the fusion site so degenerate? So they're asking the question, okay, we made the prediction.
Explain why?
Here's the data.
You know, a lot of papers, they always phrase the question and then they explain the question. Did they actually explain why? Because I'll actually go through what the explanation for this is that was predicted. So
let's pull yours up.
Yeah. Okay. Let me just. All right. So once again, guys, I won't belabor this point, but you'll see here that the banding becomes more and more detailed. So this is what it looks like, you know, when they on the diagram on the top right. This is what it kind of looks like. This is obviously they put it into a kind of like a graph, so you can actually see it much clearer. Chromosome fusion 2, you'll be able to see the chimpanzees and gorillas and humans, and you'll see basically this banding kind of aligned. You know, we're talking about 90%. It's so similar, and that's why it's, you know, that's one of the reasons why if you look at it, look at the banding, you know, one of the inescapable conclusions, and the question you have to ask is, how's it so similar? Like if you compare the other chromosomes, they're similar as well. If you compare the 2 chromosomes put together for the 1, it's also just as similar. So this is same conclusion as the previous 2 papers we talked about. It's just that the data becomes much higher resolution and you can see the banding better. I think this one is called G banding, as you can see. We started with something called B banding, R banding. This is called G banding. I have no idea what the different bandings mean. I just assume it's a different technique and a different way of staining it that gives you the better resolution, right? But this is what they call G banding. Now, some predictions, and you'll basically see this is the one at the very top here that I'm circling is essentially what Donny exactly kind of went through. This is here a kind of blown up version of the different bandings and how it aligns so that you can see it. Like, it's kind of really small to see it in the bottom right picture, blown it up so you can kind of see it. And what I mean when I say, hey, two chromosomes put together compared to human chromosome 2, the bandings align so well. You have to ask your question, why does it align so well if they're apparently two different chromosomes, right, as opposed to two chromosomes put together? So it's a question. But some predictions came out of this paper as well. So one of them is, because this is where we start to get enough high resolution to understand what the telomeres are. Before, they knew that there would be one, but now that they can actually see that there's a telomere to telomere site. So a prediction that came out of that would be, well, if we know that two telomeres kind of join together like that, what will we expect? And what we would expect is that we would see high density repeats within those telomeres, because we have, the caps of the telomeres at the end. They're pretty big, as Donny kind of said. I can't remember what it was, but we're talking about thousands, as opposed to the 798 that we see in the telomere to telomere fusion site, which is a question I'll park for a second. So Donny asked, why do we have very large telomere sites put together, but the actual fusion site's only 798 when we're talking about thousands put together? Why is it any bigger? So that's Donny's point, and it's a valid point, but I'll get to that hopefully in the next slide. But the one prediction here is, when you see it put together, we should be seeing high densities of this, because you've got two big telomeres kind of fused together. The second prediction here is that, because at this time we started to understand more about what centromeres were doing, and we now understood that two centromeres in a chromosome is very bad for the organism. So a prediction would be one of those centromeres would have to be inactivated somehow. All right, that prediction came out of this paper, and that was because we had a better understanding of what a centromere does, and we knew that having two ones is not good for the organism. Another prediction here is that, well, given that if you understand what a telomere basically protects the chromosome. It is designed to wear out as the chromosome kind of replicates. So it's actually designed to be, it's like how would I describe? It's like a padding, and the padding would itself like protect whatever it's being padded, and it would kind of wear down as it's being used. That's what a telomere does. So it doesn't do anything else than that. It doesn't code for protein. There's no gene expression. It doesn't do that. So when you have a telomere to telomere fusion, and this is me trying to explain your question, Donny, all right, it is expected that there would be a lot of degradation that happens with that, because it's not doing anything. So when you kind of fuse,
let me ask you this, Ian, just to keep it one point at a time.
Yeah.
If it was expected, did they in their initial prediction before the data came about for them to analyze and look at, did they make that prediction? Did they say, by the way, this site, these signatures should be degenerate, they should be there, but not in an abundance?
In this paper itself. Now, what it doesn't do. So what it does say is, in this paper, 1982 by Yunus and Prakash, Origin of Man Chromosome, in the science volume there, you can just basically cut and copy this. I'll send you the pack, or type it out, I guess, if you're an audience, or ask Donny for the pack, I suppose. Cut and copy, stick it into Sci-Hub or stick it into Google, and you'll get the paper. What it says are these specific predictions itself. Now, it says it would expect it. They expect it to be high density. It's expected to be degraded. What it doesn't say is we expect it to be 798. So I can't tell you a prediction that says we expect it to be significantly shorter. What they do say is we expect that it's going to be smaller or degraded from two of them added together. But it doesn't say we expect it to be that much shorter.
But then isn't it kind of subjective? Because it's like, well, we expect it to be degraded. If that's what they said, I'll take your word for it. Yeah. But degraded to them could be 10,000. It could, like, for example, this pig example, I double checked because I did a presentation on it in response to Erica. The site in the pig fusion is, take a guess. How large this is 798 base pairs. How large do you think the pig one is?
I don't know. So maybe tell me how large the telomeres were and how large was the fusion happen, so that I have a before and after kind of shot in the pig.
That's a good question. The average telomere, if I remember correctly, is somewhere between 5,000 and 15,000 DNA letters long. So let's say in the human
compared to humans, huge compared to humans for sure. Yeah.
Now, if that's comparable in the pig genome, then I think we would have an apples to apples example here. I do understand your point. We got to see the size of the telomeres in the pig genome. I think it's relatively the same, but don't quote me on that. So let's say with the human.
I don't know, Donny, and I don't
Right.
know the answer to that myself.
So in the pig genome, the fusion site is 38,000 base pairs long.
Massive compared to humans.
Massive.
For sure.
So
Yeah.
this one's 798. Now, let's say in the human chromosome 2, let's say you have these two chromosomes come together, and the telomeres, again, somewhere between 5 and 15,000. You get two that come together, so you're expecting about a 30,000 length signature, but they have to degrade. I understand what you're saying. The telomeres largely act to protect, right? They're protective end caps for the chromosomes.
Yeah.
So they degrade, then they fuse together. Okay. So with two larger telomeres coming together, you'd expect 30,000. Okay. But they would have to degrade. Okay. So let's say they degrade half that size. How come we still don't have a signature that's like 15,000 or 10,000 or 5,000 or even 2,000? It's less than 1,000. How come your pig telomeres, why didn't they degenerate? I mean, 38,000, regardless how big their telomeres are or how large their telomeres are, that's quite a big signature, you know? So I get degeneracy has to happen, but even in light of that, we'd still expect something like 5,000, even 10,000, but why 798, right? Ian,
Yeah,
go
I had to park that because my mind doesn't jump straight to making assumption that the pig telomere is not. I mean, it could be large. Like we know, like what is the size of the chromosome length between a pig and a human? It could be massive. You know, like we've got an onion. Was it an onion is what 60 times the size of the chromosome of the genetic kind of information of chromosome compared to humans. So what is it with a pig? So maybe the telomeres on the pig is very large. I don't
think
know.
the pig telomeres are roughly, because I just looked it up.
Yeah.
And it says pig telomeres are typically between 10 and 30 kilobases. And I think, so is 1 kilobase 1,000?
Yes, 1,000 base.
Okay.
Yeah.
Okay, so between 10 and 30,000 bases in length. And that's basically what a human telomere is too, I think, because human telomeres are between 15 and 30. Let's see, how large are human telomeres?
Yeah, so I've just Googled it, and it sounds like the pigs is massive. So so if you put how large is human telomeres, it says it's 5,000 to 15,000 base pairs, not kilobase pairs, base pairs. Where if you look at the pig, it says 10 to 30 kilobase pairs. So it's 10,000 to 30,000 base pairs. So it's massive.
But then here it says how big are human or telomeres in humans. It says human telomeres, for example, range in size from 2 to 50 kilobases and consist of approximately 300 to 8,000 precise or pristine repeats of CCC, TAA, TT, TTA, GGG. And so they're not that far off. 2 to 50,000. And then what'd you say the pig was? 10 to 40,000.
10 to 30. Okay. We must be looking at different data, I guess, because the one I've got is human telomeres are typically between 5 to 15 kilobase pairs, not. So
You're right. I'll show you right here. So how big are telomeres in humans? Human telomeres, for example, so there is a range in size from 2 to 50 kilobases. And then pig telomeres are typically between 10 and 30 kilobases in length. So either way, you'd expect a pig fusion. And then I'd love to look at the equid fusion sites. Because again, when I see a signature, Ian, of 38,000 bases or whatever we find in the equids, I'd like to see that data. And you have 1,000 plus tandem repeats, as in this fusion site is pristine, then the evidence is indisputable. This was a result of a fusion. But then when you look, and this is why I'm skeptical, and that's what I say, skeptical. I'm not against it because I know we both agree it's agnostic, but I don't like to jump to a conclusion just because even the majority of scientists agree with it. Because again, when I'm comparing the chromosome 2 fusion site in humans to fusion sites in animals, it's so much smaller, it's so much more degraded. And I understand your great point about how the telomeres need to degenerate for the fusion to take place in the first place. You'd still expect more though than 798, especially if you're looking at a fusion in, say, a pig
Yeah.
species of
Yeah.
38,000. Go ahead, I'm talking a
So
lot.
no. So to me, maybe it's because I'm looking at the data in a different way, but the data that you talk through doesn't disprove what's going on. So you gave me a range, all right? So um, it could very well be that we have a lower range compared to a higher range. So you could be taking the 30,000 range from the pig compared to the 2,000 range of the human. So it's still within the range that you've given me, right? One just happened to have, coincidentally, when you did the fusion, that happened at the very lower range, and then the pig happened at a higher range. And then suddenly you're comparing 2 to 30, 15 times the size. So
How do you know that?
it's
How do you
why
know the
don't.
size of the telomeres when
Why
when
don't?
the
What I'm basing is on, you've given me data points, and the
okay.
data points is based on a range. So to me, it's like, as long as it fits the range, it's possible. All right. So I
Okay,
could
so
argue
even
with
if you
you
had
to
a
say,
small and I appreciate that's a good point. Let's say you had a smaller telomeres. Okay. Sometime after the split in the human lineage, you have 2 telomeres ranging, let's say 5,000. Okay. So the smaller range. So then it's 10,000. Okay. So it would have to degrade to how, because now it's 798. What do you think the size was when the fusion happened? Well, if you had to guess.
I don't know. I mean, you're telling me that telomere size itself is ranging what? 2 to, what was it they gave me? 2 to, so it's just be an addition of whatever that range is.
I
So I think
usually say between 10 and 15, but the ranges seem to be all over the place. I mean, that source said between 2 and 50. That's a huge
yeah.
range.
So look,
So
it's hard for me. Like, as long as there's a range and it fits within the range,
okay.
it's hypothetically can work. Do you get what I mean?
Right,
And I get that the range seems quite large. So
but
yeah.
then and here's my biggest issue. And again, I'm not totally again, I appreciate what you're saying here. I'm not totally against the fusion happening. But then based on the research that Chris Roop and I did, we made a prediction. Okay, because Gutsy Gibbon put out a video saying we don't find head to head. I understand we find isolated cases of interstitial telomeric repeats. Even Dr. Tompkins pointed that out here. So if you look here, he found, this was his research, he found many instances. So here's the chimpanzee genome, here's the human genome. So he found many instances of these interstitial telomeric repeats in all the chromosomes. And he did a study on it and suggested that based on at least 2 papers from the conventional literature, that these interstitial telomeric repeats are involved in gene expression. So they're not just found at the ends of chromosomes. But to be fair, Erica made an excellent point. She put out a challenge, and Jeff Tompkins, he didn't have any data on this. He didn't have an answer to this challenge. Nobody did. No creationists did. She said, but you don't find these internal telomeric repeats in a head-to-head fashion where it's TTA, GGG, CCC, TAA, right? Finding them in mirror form, because that would suggest a fusion. So me and Chris Roop, we said, okay, we're going to predict that, yes, we do find that, but we didn't know. Nobody knew. No creationists did that project or made that prediction. So we went in, used BLAST. Chris specifically used the BLAST. He's familiar with that program. And we discovered many.
What it is, but I've never played around with it. So you'll have more experience there than me.
Yeah, it's where you can analyze the sequences between various organisms. So you can look at the human genome and you can look for specific sequences. So here's the actual BLAST analysis, right? Here's GenBank. Here's the sequences. Anyways, we found TTA, GGG, CCC, TAA in mirror form, head-to-head in many chromosomes, but in very low density, not worth showing a picture of. But in the chromosome 9, I don't want to say to our surprise, because we did predict that we'd find it, but maybe we would, maybe we didn't. That's science. But we did find, to probably the surprise of evolutionists, because if Gutsy Gibbon was saying that we don't find this. So we find, and what's interesting in the chromosome 9, so here's the section, we printed it out. you find it in mirror form. You can see it here, right? Blue and red. TTA, GGG, TTA, GGG, CCC, TAA. Here's what's amazing. Here's what's fascinating, Ian. The chromosome 9 site is more pristine than the chromosome 2 site. Now, the chromosome 2 is higher density. That doesn't mean this is not high density. This is just lower density than the chromosome 2. But the chromosome 9 area that we discovered is more pristine, right? So there's a trait here in the chromosome 9 that favors the chromosome 9. It's more pristine. The chromosome 2 area.
So I've got one question. What do you think this does in chromosome 9? What do you think this does? So the reason why I asked, I'll give you a bit of a kind of insight as to why I'm asking this question to maybe help you with your answer. But well, two things. None of this, from what I can see, none of this invalidates the prediction. So the prediction was we would expect the telomere to telomere fusion site in chromosome 2 to be degraded, because telomeres is meant to be that. It's meant to be that buffer zone. So we expect it to kind of obviously, lose CATGs or whatever the number. We expect it to lose that. So you're not going to have it pristine, because it is a function of what a telomere does. And when you look at the fusion 2 site, it's not pristine, but that's to be expected. So my question then is two things. One is, if what you found there, does it have a function? Like, does it do anything? Because when I look at those numbers, like, how many letters is that? That's 25 letters or something, if I count that stuff. So, like, how is that not just chance? And the reason why
these letters are tandem repeats, though.
Yeah. Yes.
So they're all back to back to back to back. When you go to the chromosome 2 section, right, you have one tandem here, if you can see, TTA, GGG. Yeah. But then it's broken. It's degenerate all the way up. And then you get another one.
As predicted, though, Donny. That's as predicted, though.
But how can that be as predicted if all they said was, well, we expect it to be degenerate, but they didn't really give a precise number. They didn't give any specifics. But then when we look at these animal species like the pig or the equids, I'd love to see the data in the equids. I would predict the equids. I'll make a prediction right now. The equid examples, they're going to have large signatures. And you know what? If they come back at less than 1,000, then I think that'll be a point for you.
So just to confirm, so you're going to predict that there'll be a larger fusion site, and then you're going to predict that it's going to be pristine. Will you predict that it's pristine as well? Because the pristine one is the one. The pristine will be the one that will then directly contradict the paper, because the paper didn't talk about how large it was. So I can't tell you whether that's going to be right or wrong, because the paper did not say that. What the paper said was they would expect it to be degraded. And to your point, doesn't quantify what that degradation look like. So I can't tell you, is it 5%? I can't give you that, but it will expect it to be degraded. So if you're saying that your prediction of the equine or the pig or whatever it is, you're saying that it's pristine, then that is
the pig. I know I have the data for that one, but the equids, you guys bring up the equids a lot, but I've never actually seen the data. The pig, I have the data, and I reviewed it with Ruhif, who's on your side, big fan of the human chromosome 2. And he helped confirm with me that it's about 38,000, the signature. And it is fairly pristine, because in terms of TTA, GGG, and CCC, TAA sites, you have about, I think, somewhere between 800 and 1,200 pristine sites. Okay.
Out of 38,000 doesn't sound like it's a big amount then. If you're saying that it's a 38,000. So my point here is, you have 3 billion base pairs. So that's 6 billion letters. All right. At 6 billion letters, what is the chance that 25 of them, unless you're finding this everywhere, you're finding one instances of looks like 25 letters that just out of 6 billion possible combinations, like, why isn't that just chance? Unless you're telling me there's a function, that's different. If you say there's a function, then obviously that is meant to be there. Like, it's meant to do something.
Okay, let me answer the function question then.
Yeah.
So firstly, in my technical article here that I wrote based on our findings, we found them in both forwards and in reverse in these chromosomes: 1, 2, 6, 9, 10, 15, 16, 18, 20. Your two that had the most density were 2 and 9. So that's why I have pictures here of 2 and 9. In your 2, you've only got 10 intact TTA, GGG telomere sequences. And my point about the pig is it's more than 1,000 for sure. I got to get the exact numbers. It's quite a bit. Okay, it's a lot more than 10. And I get the signature's bigger, but that's what I mean is if this was really a fusion in the chromosome 2, then you should expect, I would say, something more akin to what we find in the pig site. And I know you want to respond to that, but let me just respond to your functional question.
But just the only thing to keep in mind is what I said is expected to be degraded. And I'm just putting data because I don't know this data. So I'm putting data that you just told me in this conversation. 38,000 and 1,000. So 1 out of 38. To me, that's massive.
It could be even more. It could be even more. Remember when I was
2,000, double it. That's 2 out of 38. The degradation is still a lot. Like it's a lot.
How come the pig wasn't? So you're saying the pig fusion?
I'm using the pig.
Of
didn't you tell me the pig is
telomere?
1,000, and then you told me that the size of it is 38,000? So I'm just saying 1,000. That's that has that repeat that looks like it's pristine divided by 38,000. To me, that means at least 90% of it, 95% of it's been degraded. So to me, that still sounds like a lot of degradation. I'm using the numbers you've given me because I'm not familiar with those numbers. So I'm using what you've told me. I need to go and have a look into it. But the numbers you told me is the size of the pig is probably 38,000.
I'll send them to you after we're done.
So remember the prediction that I said was we expect that to be degraded. And what I can't tell you, Donny, is by how much, because that wasn't in the paper. So I can't tell you by what. But from what you told me, it doesn't sound like it contradicts the paper. Like you've told me, even though it's 1,000, it's more than 10. I grant you it's more than 10, which is humans, but it's 1,000 in pigs. So that's strange. It's more. But compared to the size of the telomere fusion for pigs, it's still a massive degradation, which is what the paper says would happen. So it's
okay. Let me say this. Let me say this, then we'll give you the last word because we're over an hour, and we'll be filming our part 3 very soon.
Yep.
But just because of when we're doing this, I've got another obligation, but this has been great, and I can't believe how quickly time flies by. But I'll say this: so in my article here, I do go over. So it's not only that we find telomeric repeats scattered all throughout the genome, with many of these repeats having evidence for functional activity in gene expression. So gene expression technically is the process by which DNA directs the synthesis of proteins, right? Or in some case, RNA. So it's that. So you get a functional RNA or a protein. And so these interstitial telomeric repeats are actually involved in that gene expression process. They've discovered that. So therefore, I would conclude that both the fusion site, because we do know specifically that this one's overlapped by a gene that they call the DDX11L2 gene, and in the chromosome 9 spot, which I think we would all agree is not the result of a fusion. And so therefore the question is, why do we find these repeats not only in isolation but in head-to-head, which someone like Erica said we shouldn't find? And she did have the density aspect or factor there too.
At a very small density like that.
But I wouldn't say this is a small density. See, this argument is like this. It's like if I were to say, okay, you got Mr. Smith and you got John here. Mr. Smith bench presses 500 pounds. John bench presses 315 pounds. And so John is weak. No, they're both strong. 315 and 500 are very strong and powerful bench presses. Both areas, well, let me say this. Both areas are high in density, the chromosome 2 and the chromosome 9, but the chromosome 2 is more dense. The reason why, well, that's something we can discuss. That's a good research.
So it goes down to basically, in my head, 2 things. It's like, does this sequence of TAGG, TTAGG, does that sequence that you've got highlighted in red and blue, does that do anything?
Right.
Because if it doesn't, then it's noise. Like that string of 25, I'm guessing maybe it's 30. That string of 25 to 30 letters out of 6 billion possible letter combination, to me, could be just noise. So if you say it's a function, that's a different story. If you say that actually does something, and it does something whether it's expression or protein coding, then that's different, because I have to scratch my head and go, well, that can't be random. That can't be random noise, because it's obviously functional. But if it's not functional, it lends itself to, it could be just random noise, because you're not giving me 500 letters out of 6 billion. You're giving me a very small percentage of random letters that could very well fit in 6 billion CATG kind of letters. Does that make sense? So the question is that functional or not? Because if it's functional, as I said, that will make me scratch my head. That will make me go, Well, that's really odd. That should be there then. That's a result of whether it's created there or it's fixed there. That must do something important. And it's odd that it's exactly that CATG. You know, do you get what I mean, Donny? So that if it's not functional, then it could be noise. You know what I mean?
Okay, let me say this in terms of the density argument and then also the functional point. I do have, you're free to read this. Anybody listening can read it. So I've got a whole section here in my article on our findings that addresses, or at least engages, we'll say engage, and then you can leave it up to you if it addresses it sufficiently, the density point. And then I also get into the details, how many. So we found 9 hexamers in a head-to-head array. Anyways, I want to say this and then give you the last word, and then we'll continue very, very soon in part 3. The first point you made, because I do like to leave no stone unturned, the synteny and the homology. So I wrote a detailed technical article on the chromosome 2 fusion that just engages and interacts with all the points. And so I wrote it in a way where I put the claim, right? The claim that the proponent of common descent would make. What about the similarity between human chromosome 2 and chimpanzee chromosomes 12 and 13, now dubbed chromosomes 2A and 2B? And I actually get into some of the amazing functional roles for synteny, or the 3D organization of the genomes, these the order of the chromosomes or the genes, and then also the homology point. Okay, maybe we can get that into that in part 3, but those are points also worth interacting with. Okay, so but I'm going to hand it to you.
Homework, Donny. You're always giving me homework.
Hey, you're giving me homework too. This is good.
Okay, so let me just flick through my last couple of slides, because I was hoping we'd finish chromosome fusion 2 in the part 2, but it looks like there's going to be a bit of a carryover.
It looks like the whole series might. Because
I know your audience wants you to talk ERVs. I know it, Donny, and it looks like we're just teasing the audience a little bit. Hey, well, we're going to get to ERVs a little bit later. We're going to finish this first. So
we both have very detailed mindsets, don't we? Where we want to just like take a point and just engage it to oblivion, which I love. And that's a thing I appreciate. I appreciate your fade, and I also appreciate your attention to detail, Ian.
I have a nickname for Donny. I call him the Canadian Homelander, because he he stares daggers, he blasts everything from, he doesn't like something, you're dead, basically. So
blasting evolutionary arguments, is that what you're saying?
That's the Canadian Homelander right there. So let me just flick through the couple. So the next slide is all it was, and I won't go through this because all it is that from this paper going on was 1991, we actually know the genetic code, the actual CATG, and Donny's talked about it, so I won't go into this at all. But instead of banding, we now know the exact DNA sequence that sits underneath it, and obviously that matches as well, right? Bar the discussion that we just had around sizing, bar the discussion that we just had around pristine versus the degradation and whatnot. So apart from those two that we just discussed, that the history was basically to take you guys through chemical comparison, banding comparisons, 3 different ways, each getting higher and higher resolution to pretty much the best way, which is knowing the genetic elements within that. So that all kind of points to and confirms each of the previous one. Now, this is the one I think we're going to be talking in part 3, because I heard you say that there was a functioning gene that sits across it, and I can't find anything. Right. In fact, I found a paper that said differently. So I think I'll send you this anyway, Donny, so you won't be surprised. Okay. It's the paper is listed here as well, but basically, and here's the paper here, but it does not find any. The DDX11L2 gene is not functioning. It sits in the subtelomeric fusion area, so it's not an actual telomere fusion area as well. Now, they have found...
Okay, why don't you save that one for... You know what we'll do? We'll start off part 3. Ian, you going over this slide, because I do have some slides and responses to that. We can engage. Yep. We can engage. We could...
Hey, you might have to go closer. Actually, what I'll do is I'll stick this in a private chat, so you basically have my slides here anyway, but I will be contesting with you in part 3 that DDX11L2 is a functioning gene. It is
not. Oh, you're right, because I'm saying it's a functioning gene. You're going to say
I'm saying that so my conclusion is we don't know. The paper says that there are some elements there that could be for gene expression, but it concludes that it's so heavily degraded that it's likely to be doing nothing at all. But the paper also says it sits in the subtelomere area, doesn't sit across the telomere sites. Okay, so it's right next to it. That while there might be some parts that look like it does gene expression, it's so degraded. The conclusion of the paper, it's so degraded that it's not likely. Okay, so it's not definitive. It's not likely to do anything at all. I'll send you this slide so you have it there. It's got the paper itself, so you can do a kind of copy and find it yourself. Maybe for the next one I'll do a screenshot of the paper. I didn't do the screenshot. I just did the just listed the source or whatever. So maybe I'll do a screenshot and we can read off that itself. But that's the conclusion. And the reason why I put this slide together was because I read some comment from people that said, "Oh, well, it's functional." And I can't find any papers that say it's functional. What I found is the opposite. All right. So maybe
you remember in our previous discussion I showed you from the genetic database DDX11L2 and how it had over 1,000 functional associations and co-expression. And I can send that to you if you want to get ready for.
Yeah, send that, because this paper is 2009. So maybe there's something newer. I don't know. So this is a 2009 paper by Costa Casamassimi or whatever, and it's specifically on DDX11L, a novel transcript family emerging. So it's from the human subtelomeric region. So it's from BMC Genomics. So so I'll stick to private chat and hopefully.
Okay, well, Ian, again, as always, these discussions are awesome. You make me sweat. Because these are great, technical, detailed, and thorough back and forth discussions on these significant
that's related. I like to make girls sweat. I don't really like to make you sweat, Donny, but I guess I can take that as a bit of a compliment, I guess.
Yeah, there you go. Yeah, you make me sweat because the conversations are intense in a good way. All right, Ian, we're gonna wrap this part up here, and we're gonna continue into part 3 very soon, possibly within the next 24 hours.
Fun as usual, Donny. Super fun as usual. Thank you very much, Donny, and guys listening.
My pleasure. Thanks for tuning in, audience. Leave your comments. Be nice.
Be nice.
Yeah, be nice to Ian, because this is a tamed Ian we're dealing with. God bless all.
See you guys.